So, we are going to discuss some important terms in bacteriology today, which is microbiology. तो सबसे पहले जब हम स्ट्रक्चर को डिस्कस करते हैं बैक्टीरियल स्ट्रक्चर्स को तो द फर्स्ट थिंग दैट कम्स इन आवर माइंड इज़ द अपेंडेजेस. ठीक है. तो अपेंडेजेस में वी सी के we have the flagellum and we have the pilus or the fimbria. So flagellum you can see right over here, ये है in gram negative and this is the flagellum in gram positive. It's almost similar. तो जो flagellum है it is composed of proteins और इसका function होता है motility and movement of the bacteria. सही है. Then the other structure which we have is the pilus or the fimbria. It is composed of glycoproteins, unlike flagellum which is composed entirely of proteins. Function क्या है? The pilus it mediates the adherence of bacteria to cell surface. कि जो भी बैक्टीरिया है उसने किसी भी स्ट्रक्चर पर अगर जाकर अटैच होना है तो उस अटैचमेंट में पाइलस इन्वॉल्व होगा. सेक्स पाइलस फॉर्म्स ड्यूरिंग कॉन्जुगेशन. जो कॉन्जुगेशन का प्रोसेस होता है उसमें सेक्स पाइलस इन्वॉल्व होता है. नाउ दीज वर द extensions of the bacteria, you can say. Specialized structures of bacteria, they involve the spore. It is a keratin like coat. It is composed of tryptolinic acid, peptidoglycans and DNA, right? And mostly it is present only in gram positive bacteria. और इसका सर्वाइवल कैसे पॉसिबल है कि ये जो स्पोर होता है इट इज़ अ हाईली रेजिस्टेंट स्ट्रक्चर, सो इट रेजिस्ट्स डिहाइड्रेशन, हीट एंड केमिकल्स. दिस इज़ हाउ it protects itself in harsh environments, and when the conditions are favorable, the spore just it converts again into the active form bacteria. ठीक है. अब cell envelope में there are some structures which are involved in the protection and support of bacteria. First of all, we are going to discuss about the capsule. अब इस डायग्राम में जब हम देखते हैं तो we can see कि this is the capsule right over here. Now, what is it composed of? It is a discrete layer which is usually made up of polysaccharides and rarely proteins, and it protects the bacteria against phagocytosis. किसी भी किस्म के अटैक से ये कैप्सूल प्रिवेंट करे. So it is a highly fibrous and resistant layer. Then comes the slime layer. It is just a loose network of only polysaccharides, and it mediates adherence to surfaces, especially foreign surfaces. Example, indwelling catheters. अब कैथेटर्स क्या होते हैं? देर द ट्यूबिंग्स जो कि बॉडी के अंदर मे बी ड्यूरिंग डिफरेंट काइंड ऑफ ऑपरेशंस ये बॉडी के अंदर इंट्रोड्यूस की जाती हैं. तो इनकी सर्फेसेस के ऊपर भी स्लाइम लेयर बैक्टीरिया की प्रेजेंट हो सकती है. तो ये चीज़ आपने जहन में रखनी है. We have discussed the capsule slime layer. Now we are coming to outer membrane. You can see right over here, this is the outer membrane and it is composed of some structures. We'll discuss it shortly. Now, outer membrane ke andar there are two further subdivisions. There's the outer leaflet, there's the inner leaflet. Outer leaflet ke andar we have endotoxin, which contains either lipopolysaccharides or lipooligosaccharides. And then there is embedded proteins, which include porins and other outer membrane proteins, which are abbreviated as OMPs. Right? और inner leaflet के अंदर सिर्फ phospholipids present होते हैं. अब outer membrane mostly it's present only in gram negative bacteria. और जो endotoxin है, जो structure हमने पढ़ा outer membrane के अंदर there's endotoxin present. Endotoxin के अंदर जो lipid A component है, it induces pro-inflammatory cytokines which include the tumor necrosis factor and interleukin 1. जबकि जो polysaccharide component of outer membrane है उसमें जो O polysaccharide component है, it is antigenic. It is involved in the production of immune responses. Right? फिर इसके बाद उन्हें क्या है कि मोस्ट ऑफ द आउटर मेम्ब्रेन प्रोटीन्स दे आर एंटीजेनिक, दे आर इन्वॉल्व इन मीडिएटिंग इम्यून रिएक्शंस. और जो प्रोटीन्स हैं, जो पोरेंस वी जस्ट डिस्कस्ड राइट over here, they they're involved in transport across the outer membrane. Different substances इनके through move कर सकते हैं. Now what we have discussed is that we have discussed capsule, slime layer, outer membrane, and now we are coming on to periplasm. What is periplasm? It is a space between the cytoplasmic membrane and outer membrane in gram-negative bacteria, peptidoglycan in middle. Now, it's only present in gram-negative bacteria. You can see right over here in the region between the cytoplasmic membrane and the outer membrane. This region is the periplasmic space. Sorry. इसमें क्या होता है, इसकी सिग्निफिकेंस क्या है कि इट एक्यूमुलेट्स कंपोनेंट्स एग्जिटिंग द ग्राम नेगेटिव सेल्स इंक्लूडिंग हाइड्रोलिटिक एंजाइम्स, एग्जांपल बीटा लैक्टोमेसेस. जितने भी हाइड्रोलिटिक एंजाइम्स होते हैं, मिसाल के तौर पर उन्होंने दिया हुआ है बीटा लैक्टोमेसेस, ये इस जगह पर पेरिप्लाज्मिक स्पेस में एक्यूमुलेट होते हैं. राइट. अच्छा, सेल वॉल के बारे में हम क्या पढ़ते हैं कि इसके अंदर it is composed of peptidoglycan, which is a sugar backbone with peptide side chains cross-linked by transpeptidase. And what is basically cell wall? It is a net-like structure which gives rigid support. It protects against osmotic pressure damage. So the last thing which we are going to study is the cytoplasmic membrane. Cytoplasmic membrane is basically a phospholipid bilayer. You can see right over here, this is a bilayer of phospholipids with embedded proteins, example penicillin binding proteins abbreviated as PBPs and other enzymes. Lipotecoic acid, gram positive only, extend from membrane to exterior. This is an important point to remember कि जो gram positive bacteria होते हैं उनकी cytoplasmic membrane में lipotecoic acid present होता है only in gram positive. Now, cytoplasmic membrane, it is a site of oxidative and transport enzymes. The penicillin binding proteins involved in the cell wall synthesis, they're also present right in the cytoplasmic membrane. और जो लिपोटिकोइक एसिड्स जो कि ग्राम पॉजिटिव बैक्टीरिया में प्रेजेंट होते हैं, व्हाट डू दे इंड्यूस द ट्यूमर नेक्रोसिस फैक्टर अल्फा एंड इंटरल्यूकिन 1. सो दिस इज़ a general overview of basic bacteriology in which we have just discussed about the bacterial structures, in which we studied about the appendages including the flagellum and the pilus or fimbria, the specialized structures which involves the spores, and the cell envelope which is composed of capsule, slime layer, outer membrane, periplasm, cell wall, cytoplasmic membrane. And this is a diagram which if I just try to zoom it, you can clearly see the difference between the gram positive and the gram negative bacteria. This is gram positive and this is gram negative. So I think that's it for today.